So now we've talked all about how the brain develops in the early stages. We said how neurons are born, how they differentiate, how they migrate to their final location. We talked all about how axons that are growing through the growing brain find their way and figure out where they're supposed to grow to, how they form synapses, and stuff like that. But that's not the end of the story with brain development. In fact, there's a lot that happens after all the neurons and the connections that have been are formed in the brain that happens through development and to some extent all the way through the lifetime of the individual. One of the important things that happens later in development is many of the neurons in the neural tube that were formed even after they've differentiated, even after their axons have grown into their target region, actually die. So it turns out that that more neurons are actually created in the developing brain, in the neural tube, than are really needed in the adult brain. And what happens is these neurons, these excess neurons, they send axons to their target location. But if more input neurons send axons than what are available in the target location, then only some of those neurons will form synapses. So this neuron here, and this neuron here, and this neuron here, when their axons get to the target, they have nothing to connect to. There are no neurons here for them to synapse with, and in addition, most neurons have a sort of limited synaptic capacity, meaning that even if they could form synapses with the neurons that are in the target region, those neurons can only form so many synapses. So if they get there late, or they just target the wrong part of that structure, they just won't find any postsynaptic cells to form synapses with. And that causes those cells to die. So later on, those neurons that didn't form synapses will die off. But they don't die just uncontrollably. So when a cell dies, normally due to disease or damage or exposure to toxins or whatever, they die through a process called necrosis. And necrosis is basically an uncontrolled cell death, where the cell bursts open and all the cytoplasm goes everywhere, and that's bad. Death by necrosis is bad, obviously for the cell, but it's really bad for the surrounding cells, because it turns out that some of the things inside a cell are actually toxic to other cells. For example, things like potassium. So we've already talked about how potassium has a higher concentration inside the cell than outside, and that's important for maintaining the membrane potential. So if you break open a cell, the cytoplasm becomes the extracellular fluid for surrounding cells, and that can disrupt all the various ion concentrations and expose the neighboring cells to, again, things that would normally be inside the cell and can be toxic. So instead of dying through necrosis, these cells die through a process called apoptosis. Apoptosis is comes from a Greek word that means to fall off or fall away, because it's meant to evoke the ideas of trees falling off of leaves falling off of a tree in the fall. So whenever it gets to be fall, the leaves turn brown and they fall off the tree in a timely and more or less orderly way. And that's how cells that die through apoptosis experience cell death also. The cell goes through several stages. It's very tightly controlled. The DNA gets chopped up into pieces. The organelles get sequestered into little vesicles. The cytoplasm gets sort of gradually removed or packaged into those vesicles. And then the vesicles can be recycled by microglia or by adjacent cells. But the idea is that the cell dies in a way that's not toxic to its neighbors. And the reason that the cells die, again, is not just because they can't form synapses, but specifically because they don't get to be exposed to special proteins called neurotrophins. So neurotrophins are an example of a type of signaling molecule called a trophic factor. So the word trophic means to grow or to thrive or having to do with growth. And so a trophic factor is a protein released by a cell that promotes the growth and survival of other cells. So it turns out in many parts of the brain, there are neurons or glial cells that release trophic factors, but they only release so much. So if you have this situation where you have a whole bunch of neurons sending axons to a target region that has not enough neurons to support it, then those neurons will only get exposure to so many of those trophic factors. So these trophic factors, again, are proteins that presumably are being expressed by or nearby these target neurons. And so when the first axons get there, they are supported by those trophic factors. And then when the later ones get there, or if the ones that don't end up in the right spot arrive, this is also a mechanism, sort of an error correcting mechanism. So if an axon grows by mistake into the wrong part of the brain, again, it would not get the necessary trophic factors to stay alive. And that's a way to sort of fix wiring errors in some cases. But the trophic factor, it's worth mentioning, is not some sort of nutrient or some sort of metabolic factor that the cell needs to live. The trophic factor is really just a signaling molecule. It's a protein that binds to a receptor on the input neuron and basically stops the apoptosis process. Apoptosis, in a sense, is almost like, we call it controlled cell death, but sometimes it's called or thought of as cell suicide. And so that's actually what trophic factors do. They don't so much keep the cells alive as they do stopping the cells from committing apoptosis. They stop the molecular pathway that would otherwise lead to apoptosis. And so this is, again, not an accident or not an uncontrolled system. This is something that's sort of programmed into the cell's life cycle. And this is not just important for neurons, of course. Lots of cells and lots of tissues are capable of apoptosis. And the other reason that apoptosis is important is that cancer cells, of course, are cells that divide uncontrollably. And many types of cancer are caused by mutations in genes that otherwise would be related or trigger apoptosis. So many cancer cells lack the ability to go through apoptosis, so they just keep dividing and growing. In fact, and some of the research into cancer or tumor biology anyway is to try to reset those mechanisms, try to convince those cancer cells to kill themselves, to protect themselves, to protect the individual. Of course, with if you're studying neuroscience and you're interested in apoptosis, you might have the opposite approach, because the fact that cells die during development, of course, is necessary, but it also means that the absence of trophic factors can be a problem in after brain injury. It's one of the things that can prevent regeneration or preventing neurodegeneration in the brain. And so neuroscientists are interested in apoptosis for the opposite reason. Tumor biologists want to kill cells, neuroscientists want to keep them alive, but they both are interested in apoptosis just for different reasons. Now, another problem, in addition to too many cells in the brain, is too many synapses. So, like I said, when an axon grows into its target location, there are guidance cues that tell the growth cone where to go, where not to go, and then there are molecular cues on the target neurons that trigger synapse formation, and or trophic factors that allow incoming neurons to survive. But you still end up with extra connections. You have connections between neurons that aren't really appropriate for the function of that circuit. So, for example, let's say we have inputs from the eye to the thalamus. So that's what's going on over here. So here's the different eyes. Let's say this is the right eye. And let's say this is the left eye. And so these neurons, these cells here, are retinal ganglion cells. And those retinal ganglion cells form axons that go to the LGN of the thalamus. And if you recall, the LGN is divided into layers, and only some layers get input from one eye and other layers get input from the other. So presumably these are two different layers of, let's say this is layer 1, this is layer 2. I don't know which layers those are supposed to represent, but it doesn't really matter. So ideally, the input from one eye would go to one layer, and the input from the other would go to the other. But what actually happens is some axons just mistakenly go to the wrong layer. So here we've got this axon is a branch of one of the left eye retinal ganglion cells, and this axon is coming from the right eye, and they're both going to the wrong layer. They're not supposed to be in that layer, forming synapses, but they do anyway. So they form synapses. It's presumably within the synaptic capacity of those neurons. They're getting trophic factors, so they're surviving. But now we've got these inappropriate connections. So there has to be some mechanism to get rid of those connections, because that's eventually what happens. Eventually, those errant axons retract and are essentially pruned back. So there are a number of mechanisms that control this or allow this to happen. One is something called Hebbian modification. And it's just called that because it was initially postulated by a guy named Donald Hebb, and it has since been established. But Hebbian modification basically is the idea that if you have two neurons that target the same postsynaptic cell, one of the neurons, if either of the neurons is active at the same time as the postsynaptic cell, that synapse between those cells will become stronger. Whereas if you have two neurons or another neuron that's connected to a postsynaptic cell, and that neuron is active in a way that doesn't correlate at all with the presynaptic cell, then that connection will eventually be lost. And so in the case of the eye, the way this works is you have each eye projecting to both layers. And of course, this has been studied in mammals and animals that go through an extensive embryonic developmental stages. And so, of course, this is at a time when these animals, the retina has developed, but there is no light. So the eyes aren't seeing anything. But it turns out that retinal ganglion cells and other neurons in the nervous system experience a lot of spontaneous activity, meaning they just sort of randomly fire. And this happens in both eyes, but most of that spontaneous activity is correlated within the eye. So what that means is if one of these cells from the right eye is active, then other cells in that eye will be active at the same time. So they generally experience these bursts of essentially random activity, but that activity is connected because, of course, the retinal ganglion cells are connected to each other by horizontal cells and so on. So when they experience this, they do it at the same time, which means that when the inputs from the right eye are active, they're going to simultaneously activate the neurons that they synapse the most on, which is, in this case, the neurons in LGN layer, what we're calling layer 2. The input from the right eye to layer 1, there will be some activity here, but because this neuron is active by itself, it's not getting as much input from the left eye. It will have a much weaker effect on this cell. And so the same thing presumably would happen in the left eye. So when the left eye retinal ganglion cells are spontaneously active, they're spontaneously active together. Their activity is correlated, in other words. And so the layer 1 neurons in this case will be active because of that piling on of activity. So again, even though these synapses, so again, these are our inappropriate synapses. Oops. Here, these are our inappropriate synapses. They're not supposed to be there. Even though they are active, their activity is not correlated with the other inputs to that eye. And so eventually what happens is two things actually happen. Those synapses retract. They eventually become weaker and weaker and just disappear. And then the connections between the correct eye and the correct layer become stronger. So what you eventually see is these neurons start to form new connections in the correct layer. Another way to say this, the kind of way to remember it, is neurons that fire together wire together. So these neurons from the right eye that are firing together, meaning that their activity is correlated, they're firing at the same time. The cells in layer 2 that they both synapse on are experiencing that activity simultaneously, and those connections become stronger. So again, they're firing together. This is firing together, and then this is wiring together. That's what Hebbian modification does. If you have strong correlated activity of two neurons on the same postsynaptic cell, then both neurons experience stronger or subsequently experience stronger synapses. Whereas if you have two neurons synapsing on a third neuron and their activity is not correlated, or whichever one of them is not correlated, that neuron's connection will become weaker. In fact, the corollary for Hebbian modification is neurons that fire out of sync lose their link. But it's just a way for these connections to gradually rearrange themselves based on correlated activity. So this is why each layer in, for example, the LGN only gets input from one eye and not the other, because the input from those eyes is correlated. So what actually there, and then on top of that kind of rearrangement, there are also a lot of examples where synapses can change their strength over time. This is called synaptic plasticity. So we've talked about plasticity before. We talked about cortical plasticity, but cortical plasticity is really just a special type of synaptic plasticity that happens to occur in the cortex, the cerebral cortex. Plasticity, though, can happen in any part of the brain and does happen in every part of the brain. And so synaptic plasticity is just a change in synaptic strength. But first of all, what do we mean by the term synaptic strength? And basically, it's pretty much what it sounds like. Synaptic strength, or the strength of a synapse, is a measure of how effective it is, meaning how likely is it that the presynaptic neuron will be able to create a response in the postsynaptic neuron. So a weak synapse, in other words, when it releases its neurotransmitter, will create either a very weak response in the postsynaptic cell or no response at all. And you can measure that. You can measure strength as the size of an EPSP. So remember, an EPSP is the degree of depolarization in the postsynaptic cell. Or you can measure it as the number of action potentials that are produced in the postsynaptic cell. Of course, that's how you define synaptic strength for an excitatory synapse. An inhibitory synapse, you would define strength as its ability to inhibit an excitatory signal. But in any case, it's just how good is that synapse at creating a response in the postsynaptic cell. And so again, you can measure it by this way. So here, what they're doing is they're applying an electrical current to the presynaptic neuron, or to the axon from that neuron, which is, you assume, causing this neuron to release neurotransmitter, and then you record the membrane potential of the postsynaptic cell. And then you just look to see how large is the EPSP, or how many action potentials does it produce, and that's how you measure the strength of that synapse. And it's pretty easy to show that that synaptic strength can change. This can be done artificially. So here what they've done is applied what they call a conditioning stimulus. Basically, what that means usually is from this presynaptic electrode here, they just apply a strong so a picture of a lightning bolt. They just apply a strong electrical shock to that electrode, and that presumably causes that axon to release a whole bunch of neurotransmitter. And when they do that, that in many synapses will cause the postsynaptic cell to respond more to larger or respond in greater magnitude to subsequent stimulation. So here again, what they're doing here is measuring the baseline activity of this synapse, and to do that they apply kind of a moderate shock. So they're kind of weakly shocking this synapse during this phase, just to get an idea of its baseline activity. And then during this phase, they apply a conditioning stimulus, which is a very much stronger electrical signal, and then go back to doing the weak electrical signals just to measure the response. And what you're seeing here is pretty quickly, actually, an increase in the response in that cell that is sustained. So they measure the baseline for about 15 minutes, apply this conditioning stimulus over a very brief period of time, like maybe just a few seconds or a few minutes, and then come back and record or measure the synaptic strength again. And what you're seeing here is there's an increase of almost 50% of the response of that cell that lasts for a long time. So it lasts all the way to 30 minutes. So up to 30 minutes later, this cell is still has an increased response as it did when it from what it experienced from the baseline. So we call this increase in response potentiation. And that's just a word that means there's an increased response from that cell, and because it lasts a long time, it lasts at least 30 minutes. Sometimes you can see this kind of effect for days after you've done the conditioning stimulus. It lasts a long time, so we call this long-term potentiation, or usually it's just called LTP. So this is LTP. It's just an increase in the response of a postsynaptic cell to some sort of strong conditioning stimulus. And so long-term potentiation is one of the mechanisms that, for example, drives Hebbian modification. So, in this case, we're talking about an artificial setup where someone's manually electrocuting this little presynaptic cell. But in, for example, the projection to the LGN, the conditioning stimulus is just this highly correlated activity. So as long as this neuron is active at the same time as those inputs, that increased activity presumably could be one of the things that could trigger LTP. And LTP, so LTP is an important mechanism for driving changes in synaptic strength. You can tell by the fact that it lasts so long that it must be a physical change in the synapse, meaning it can't just be, say, a change in ion concentration or something else, some electrical signal factor in the postsynaptic cell. It must be something where the actual physical structure or the proteins present in the cell are changing. And so there are a couple different mechanisms that can cause LTP. Probably the best studied one is through our old friend, the NMDA receptor. So you probably remember, hopefully, that NMDA is one of the receptors that is activated by glutamate. So glutamate is the most common excitatory neurotransmitter in the brain, and when it is released, it can activate multiple types of receptors. One of which is the NMDA receptor, the other of which is the AMPA receptor. And if you recall that, then you probably remember that the AMPA receptor is a sodium, a glutamate gated sodium channel. So when it is activated by glutamate, it lets sodium in. That causes depolarization of the membrane. And if that depolarization is strong enough, it will create an action potential. Whereas NMDA receptors are also opened by glutamate, but they are also blocked by a magnesium ion. So that magnesium ion prevents the NMDA receptor from actually letting in any ions unless it goes away. And so when a cell is at or close to resting potential, so here over here on this side, we've got a cell that's at resting potential. Even though some sodium is coming in, if there aren't enough of these receptors, or if the number of active open channels is not great enough, that depolarization won't be enough to allow the NMDA receptor to become active. However, if something causes this postsynaptic cell to become depolarized enough, then that magnesium ion will leave. Glutamate, if glutamate's present, the NMDA channel will open, it'll let sodium through, but more importantly, it lets calcium in. And remember that calcium is an ion that triggers a whole bunch of downstream effects inside cells. And it's that downstream effect, or those downstream effects of calcium, that presumably can trigger the change in the synapse. So, so these are kind of different ways of looking at it. So again, let's say we've got this situation over here on the left first, where before LTP is induced, we have three different synapses here, some of which have some NMDA receptors and some don't. I mean, they all have NMDA receptors, but some have also with their NMDA receptors are AMPA receptors. So these are our AMPA receptors in blue, and these are our NMDA receptors in pink. So the AMPA receptors are present on some synapses, but not others. So when this cell releases glutamate, when these presynaptic terminals release glutamate, this cell in the middle will be active. I'm sorry, this synapse in the middle will be more active because it has AMPA receptors, but these synapses out here won't really respond to glutamate. So in other words, these are relatively weak synapses because they don't have AMPA receptors. They're only responding or can only respond with NMDA receptors, but if the postsynaptic cell is not depolarized enough, these NMDA receptors won't really do anything. But then during LTP induction, so while we're just really blasting these cells, the presynaptic axons, with electrical current, so these presynaptic terminals are releasing a bunch of glutamate. So we just are flooding this postsynaptic cell with glutamate. It's going to activate both the AMPA and the NMDA receptors simultaneously. And the depolarization caused by the AMPA receptors being activated is going to be sufficient to trigger calcium release through the NMDA receptors, even at those synapses where there are no AMPA receptors. So the depolarization from these cells, remember, will travel to the other synapses in that cell. So this is that EPSP that's moving throughout the cell, and it doesn't have to go very far to go to just the adjacent synapses. So when that EPSP gets to those other dendritic spines, that will, along with, again, the glutamate that's also present, will allow those NMDA receptors to become active. So they let calcium in. And then when calcium comes in, again, it has a whole bunch of effects. One of the things that calcium can trigger in these neurons is it can cause AMPA receptors to be embedded in the membrane. So it turns out that a lot of these neurons have AMPA receptors that are not actually in the extracellular or in the cellular membrane. Instead, they're packaged into little vesicles. So these little vesicles can be hanging around inside the dendritic shaft or inside the cell body. And when calcium is released from, or there's a calcium influx into these spines, that can cause these vesicles to be moved. They can actually translocate from the dendritic shaft into the dendritic spine, and those proteins then get embedded in the membrane. So now you have this situation over here where all of these synapses now have an increased number of AMPA receptors, which means that all of them now will have a greater response to glutamate. And so this is what allows LTP to happen. Even hours later, if you activate these axons again, these synapses will respond. And so that is presumably what would be happening in these Hebbian synapses. So the new synapses that form and the existing synapses that become stronger are being strengthened by perhaps a mechanism like this. There are other mechanisms that can cause LTP, but this is probably the simplest one. Now, of course, the other half of the Hebbian modification coin, other side I should say, is that neurons that fire out of sync lose their link. So if you have two neurons, or a neuron that's forming a synapse with another neuron, and the activity of those two neurons is not correlated, then over time that connection will become weaker and can eventually disappear. And when that happens, when actually you see a decrease in the response of a cell to stimulation from a presynaptic cell, that's called long-term depression, or LTD. And so LTD works through actually a similar mechanism to LTP. So over here, now we've got, again, a dendrite forming now 6 different synapses with 6 different inputs. And so on one side, you have correlated activity. So these 3 synapses over here are correlated, which means that they're all, when one of them's activated, for the most part, they're all 3 activated at the same time. So if they're all activated at the same time, then they will trigger those NMDA receptors to be active, because glutamate will be present and the membrane will be depolarized at the same time. Meanwhile, if you have uncorrelated patterns, meaning basically noise, so these 3 cells over here in blue are active, and perhaps even active to the same degree as these yellow ones, but their activity is not correlated. So in other words, it's basically random. So whenever an action potential gets to one of these synapses, it has no relationship whatsoever to the presence of an action potential in one of the others. So that means that glutamate is released at these synapses and maybe activating some AMPA receptors, but the amount of depolarization is not nearly enough to activate very many of those NMDA receptors. Now there has to be some NMDA, but it turns out there's sort of, because calcium, it turns out, is necessary for LTD to occur, but there's a certain threshold of calcium necessary to sort of switch from LTD to LTP. So high levels of calcium coming in will trigger LTP. Low levels of calcium will trigger LTD, which is basically the opposite mechanism. You have AMPA receptors actually being internalized, which means they're kind of removed from the membrane, where they can't be used anymore, or they can't respond to glutamate. And so again, that's due to uncorrelated activity. Now, of course, no calcium would mean there's no activity. So you actually have to have some activity for LTD to occur. It just has to be random or uncorrelated activity. But again, this is just another mechanism. This presumably is how, back in our example up here, how these aberrant synapses would be removed, because this neuron here, this neuron from the left eye, its activity is not correlated with the activity from the right eye. Its connection would become weaker and weaker over time, and it would be it would eventually, even, it may be like this at first, where the synapse is still there just aren't any very many receptors. But over time, synapses that are very weakly correlated or aren't active at all eventually just retract. This, by the way, again, and we may talk about this more later, this is another mechanism. This is an important mechanism, not just in development, but in plasticity throughout lifetime. And so we've talked about examples of plasticity in the context of the somatosensory cortex and the motor cortex, but plasticity happens throughout the brain. at pretty much all stages of life. And in fact, this kind of cortical or a synaptic plasticity or synaptic changes in synaptic strength also underlie a lot more day-to-day activities of the brain, including memory. So later on we'll talk about how memory works, but this ability for synapses to change their strength is critical to that. It's also worth pointing out and mentioning that, again, like we said, neurons that don't fire in this correlated way or don't fire at all eventually lose their connections. In fact, there's a lot of evidence that there's sort of a use or lose it property to a lot of circuits in the brain. If you're not using or if a part of your brain is not active, if a synapse or connection is not being used, eventually those synapses become weaker and weaker, and those connections disappear. And there's evidence, in fact, that people who are not active. So one of the things that can help deal with things like dementia as people age is keeping up mental activity. So people who stay mentally active all the way into old age, they read a lot, or they travel a lot, or they do whatever hobbies they're into, as opposed to those people who, as they age, become more and more sedentary. They tend to show better outcomes in terms of dementia. And one of the things that could help with that is, again, just keeping those parts of your brain active, which prevents these synapses from getting lost. And I think that is the end of that chapter. So again, thanks again for listening.